Okay, so welcome to this next video in the playlist on synaptic mechanisms. In this video, what we're going to talk about is endocytosis. Okay, and specifically what we're going to focus in on in this video is the endocytic motif. So we're going to have a series of videos in which we're going to discuss endocytosis, and we're going to go through each of the portions of endocytosis in tandem, basically. Well, not in tandem. We're going to do them in sequential order, basically. Okay, so what I'm going to do in this video is we're going to firstly discuss an overview of endocytosis, an overview of what happens and why we need it in axon terminals. Then what we're going to discuss is endocytic motifs, and then we're going to discuss a bit of experimental background, i.e. how endocytic motifs were first discovered. Okay, right, so let's start with the motivation. Okay, so we're talking about synapses, so we'll draw an axon terminal here, okay, and we know that when an action potential propagates down the axon of this neuron and reaches the axon terminal, what it will cause is it will cause the fusion of the vesicles which are docked with this presynaptic membrane. So here are these vesicles which are docked with the presynaptic membrane, the so-called readily releasable vesicle pools. So let me just write that down, readily releasable vesicle pools. So this is the name for those synaptic vesicles, which is just the name for vesicles which are filled up with neurotransmitter. And this is the name for those synaptic vesicles which are attached to the membrane by protein complexes. And now let me fill up their lumens with neurotransmitter. Okay, now, when an action potential arrives in the axon terminal, what happens is these vesicles, some of them will fuse with the plasma membrane. Okay, so we're going to put in membrane from the synaptic vesicles into the plasma membrane. Okay, so let's have a look at this. So here is our axon terminal here. Okay, and what you're going to get is you're going to get fusion of the synaptic vesicle with the plasma membrane. So that now means that the plasma membrane has a greater surface area because you have put the membrane from, let's say, one of these synaptic vesicles. So let's have this synaptic vesicle in orange. Let's say this synaptic vesicle is fused with the membrane. Okay, so you have now added the phospholipid bilayer that was making up the boundary of the synaptic vesicle. You've added that to the plasma membrane. Okay, so what's going to happen over time is you're going to put more and more of these vesicles into the plasma membrane. The surface area of the axon terminal is going to get bigger and bigger and bigger, and you're going to run out of vesicles. So what we need to do is we need to now discuss the recycling process. We need a way to take the membrane back out of the cell membrane and put it back into vesicles, basically, and then refill those vesicles with neurotransmitter and recycle them, basically. We need a process of recycling. Okay, so what we're now going to do is discuss this process of recycling, and that process of taking membrane from the cell membrane and pinching it off to form a vesicle, that's the process of endocytosis. We're now going to discuss endocytosis. Okay, so the first thing to discuss is that endocytosis in axon terminals is triggered when you have exocytosis. So when you fire action potentials along the axon and those action potentials reach the axon terminal of the neuron, then what you're going to get is you're going to get fusion of these docked synaptic vesicles with the presynaptic membrane. But at the same time, what's going to be happening, let's say over here maybe, is you're also going to be endocytosing membrane out of the plasma membrane. So when you release a bunch of synaptic vesicle membrane into the plasma membrane, you also activate the process of recapture of this membrane back into synaptic vesicles. So when you activate exocytosis, you also activate endocytosis. So when an action potential, which I'll abbreviate to AP, propagates into the axon terminal, we know that this causes calcium to go up in the cytoplasm of the cell. So calcium goes up, and this is what triggers the exocytosis of the docked synaptic vesicles with the presynaptic membrane here. Okay, so it causes vesicle fusion and the release of the neurotransmitter contents into the synaptic cleft. But at the same time, what it's going to trigger is endocytosis, and we can show that it is the calcium signal that is going to cause endocytosis via a toxin that we get from the black widow spider. Okay, so the black widow spider is a very poisonous spider that you find, I think, in places like Australia. So where should I write this? I'll put it up here. So, black widow spider. Black widow spider. It has a venom, basically, that is extremely toxic. Black widow spider. And the full name for the black widow spider, just for interest, is Latrodectus mactans. Latrodectus, there we go, fixed it in, mactans. Okay, right. And this spider produces a toxin in its venom known as alpha-latrotoxin. Okay, and for short, alpha-latrotoxin is often abbreviated to alpha-LTX. So L for latro, TX for toxin. Now, alpha-latrotoxin's mechanism of action is not well understood. However, what happens is when you expose a neuron to alpha-latrotoxin from the black widow spider venom, then what's going to happen is you're going to get massive exocytosis of the docked synaptic vesicles. Okay, so when we put our alpha-latrotoxin onto our axon terminal here, so it goes into the cytoplasm, and somehow, by mechanisms that are still controversial, what it causes is massive exocytosis. It causes all of these docked synaptic vesicles to fuse with the plasma membrane and exocytose their neurotransmitter contents into the synaptic cleft. Okay, now the important thing is that it causes this exocytosis without calcium. It does not cause a rise in calcium. So when you poison an axon terminal with alpha-latrotoxin, all of the docked synaptic vesicles fuse with the plasma membrane, but you do not get a rise in calcium level. So it's not merely acting by raising calcium level in the axon terminal. Okay, right, so that means we can produce exocytosis without producing the calcium signal. And basically, if you do this, what you get is a huge amount of exocytosis, but you do not get the activation in endocytosis. Okay, so it's not merely the process of exocytosis occurring which triggers the process of endocytosis to occur. Instead, it appears to be that when calcium goes up in the cytoplasm of the cell, it both causes exocytosis of the docked synaptic vesicles and also endocytosis of membrane from the plasma membrane of the cell in order to make new vesicles. Okay, right, so that's a little experiment that can show that. Okay, so what we now want to do is just give an overview of the endocytic process. Okay, so a rough overview, which we're going to go into in much more detail. So a rough overview is here. So, if this is the phospholipid bilayer here, let's say we have some protein, which we'll put here. So this line that's straddling the membrane, this represents our protein, which is in the membrane of the cell, which we want to endocytose, basically. So you can imagine that maybe it's synaptobrevin 2, or maybe it's synaptotagmin, because when we exocytose these synaptic vesicles, we fuse the synaptic vesicle membrane with the plasma membrane. Now we know that in the synaptic vesicle membrane there are synaptobrevin proteins and also synaptotagmin proteins. Okay, so when we fuse the synaptic vesicle membrane with the plasma membrane, what we're going to end up with is these two proteins, synaptobrevin 2, I'll label them up. So this is synaptobrevin 2 here. Okay, and synaptotagmin, generally 1 or 2, is involved in neurotransmitter release. Synaptotagmin 1/2. Okay, these are going to now be in the plasma membrane, so you can imagine it's one of these. Okay, and they're going to need to be re-endocytosed. Basically, we want to when we re-endocytose the membrane here, we want to also capture these proteins back up because they are essential for synaptic vesicle docking and also synaptic vesicle fusion, so we want them to be taken back up when we take back up this membrane. So you can imagine it's either synaptobrevin 2 or synaptotagmin 1/2. Okay, so this is our target protein basically. Now, what's going to have to happen is some sort of protein is going to have to recognize that the target protein needs to be re-endocytosed. So this is what's known as an adaptor protein, and we'll look into these in a lot more detail. But for now, I'll just draw it like so. Okay, so some adaptor protein is going to need to come and bind to the cytoplasmic domain of our target protein, which you can think of as either the cytoplasmic domain of synaptobrevin 2 or synaptotagmin 1/2. So this is an adaptor protein that comes and recognizes the cytoplasmic domain of the target protein and recognizes that this target protein needs to be endocytosed, basically. Then, what needs to happen is we need to basically bud off the membrane. So we need the membrane basically to bud off, form a little invagination like so. Okay, so that we can form or begin to form our vesicle. Okay, so this is what's going to be our vesicle here. So here's our target protein, here's our adaptor protein still bound to our target protein. Okay, and we're starting to form the vesicle here. Okay, and this process of the invagination of the membrane like that into an almost vesicle-like structure, this is what's known as vesicle budding. So this is vesicle budding. Okay, right, and then finally what we need to do, and there's going to be a protein involved in budding this, and that protein is going to be clathrin. So clathrin is going to be responsible for this, but we will look at the structure of clathrin and how it achieves this budding in a separate video. Okay, and then finally we need to pinch this vesicle off. So we go from this to this. We need to cut this sort of connection that connects this bud with the plasma membrane. So we need to pinch this off basically. So what we need to happen is we need it to go like this and it to narrow basically at this starting point here. Now which one is this? This is the inner leaflet. So here's the inner leaflet, here's the outer leaflet. Okay, so we need this pinching off process so that it actually closes it up, and then we'll have a synaptic vesicle. So this is the pinching off process. And again, there are two proteins which are involved in this, and they are dynamin, which again we'll have a separate video on, dynamin, and also endophyllin. Endophyllin. Right. Okay, and then finally what we'll do is we'll have our endocytosed vesicle. So here's the plasma membrane, and here is our endocytosed vesicle. Okay, right. So, and it will have our target protein in here. Okay, so in the next video, what we now want to look at is this first process here. How does the target protein get recognized by adaptor proteins? What signal does it have on its cytoplasmic domain that tells the adaptor protein, look at me, I need to be endocytosed? And that sequence on the cytoplasmic domain of the target protein that tells the adaptor protein that it needs to be endocytosed is what's known as an endocytic motif, or motif. Don't know quite how I went to motif. Endocytic motif. Okay, which is the topic for this video. So we're going to now look at the endocytic motif. We're going to look at that in more detail in the next video. And I just want to say one last thing, that this sort of a vesicle is known as a endocytic vesicle. It is not, repeat, it is not called an endosome. Endocytic vesicle. Okay, the endosome is a different thing, but people often call these endosomes. They are not endosomes. The endosome is a much bigger structure than an endocytic vesicle. Okay, so an endocytic vesicle is not an endosome, and it's endocytic vesicles that you form when you go through the process of endocytosis, and we'll talk about endosomes in later videos.